Good morning, students. The lesson that we are going to begin today is sexual reproduction in flowering plants. So this is the second lesson of your NCERT book. We have already completed the first lesson, that is reproduction in organisms. So let's start this lesson. What does the topic talk about? It talks about the process of reproduction in case of flowering plants, and whenever we are talking about flowering plants, we are talking about angiosperms, right? So flowering plants boli kole flower thaka plants bilakok ami kuntu group ot thoisu, angiosperms ot thoisu. So sexual reproduction in flowering plants, reproduction boli koa loge loge we have to talk about the reproductive part of a plant, right? So what is the reproductive part? It is obviously the flower that this entire chapter revolves around, right? So as far as the morphology of the flower is concerned, we have already discussed it during the chapter called as morphology, right? So what we will be discussing here is only the reproductive part, right? So starting from or talking about sexual reproduction, so first we are concerned with the part or the reproductive part, that is the flower. So when we talk about the flower, we remember something like this. Some of the basic informations that we remember, if suppose we are talking about a flower like this, we know a flower is made up of 4 holes, right? And what were the 4 holes? So just to have a brief idea, so if this is the flower, then this is called as the thalamus, right? This is this calyx, this one corolla, androecium, and gynoecium, right? So this is the gynoecium. So how does a flower look like? Almost a flower looks like this. So it has 4 holes, and what are the 4 holes? Calyx, corolla, androecium and gynoecium. If we talk about calyx, what is the individual part called as? It is called as sepals. In case of corolla, it is called as petals. Androecium, stamen, and gynoecium is carpel, right? Out of these 4 holes, what we know that these 2 holes are considered as essential holes, and these 2 are called as accessory holes, right? So a flower has 4 holes, and out of these 2 essential and 2 accessory. Now what we understand by essential and accessory, why are the 2 holes, or this one and this one, androecium and gynoecium, is said to be essential holes? Because we know what is the main function of a flower, obviously to reproduce, right? So for reproduction purpose, generally kuntu organ, which part of the flower is most important? It is always the male part, and the female part of the flower that is more important. So we have the male part, that is the androecium, and the female part, that is the gynoecium. They are the most important parts of this flower as far as reproduction is concerned, and so they are considered as essential holes. Next we have something called as accessory holes. What is accessory? Accessory means what? Helping, right? So if you are talking about calyx and corolla, what do? They help. Help in what respect? Help, for example, if you are talking about helping, the most common thing that we talk about is attraction, no? Flowers bilakot insects bilak attract hobo lagibo, attraction or karne the different colors of the petals are most important, right? And once they get attracted, it leads to pollination, and ultimately that's a source of reproduction. So these are accessory holes and these are essential holes. In this chapter, we are not going to talk about in details about calyx and corolla, because we have already discussed during the morphology class. Here what we are going to talk about is about androecium and gynoecium. That is, how does the reproduction, or how does the formation of pollen grains, or how does the formation of the egg takes place, right? So coming back to this, so essential, accessory we have understood. Next is, we will be talking about the first part or the male reproductive part of a flower, that is androecium, right? So androecium and gynosium আমি ইয়াতে কি কৰিম details পঢ়িব লাগিব তাকো we are not going to talk about the morphology but we are going to see the process how the egg and the pollen grains are formed. So when we talk about androecium কি হয় male reproductive part no তার individual member টোক আমি কি বুলি কম stamen. Right? Some informations about stamen. What is a stamen or who forms a stamen? The stamen is nothing but a modified leaf. It is also called as microsporophyll. Right? A stamen is a modified leaf. It is called as microsporophyll and it consists of anther and filament. So we have two parts of a stamen, that is the anther and the filament. Now, there are certain things to remember from or to understand from this two parts. First, what we have said, what is a stamen? A stamen is a modified leaf. Now, there is a difference between these two statements. If I say a stamen is a leaf and if I say a stamen is a modified leaf, এই দুইটা সেন্টেন্সৰ মাজত ডিফাৰেন্স এটা আছে and what is the difference? Leaf or modified leaf, stem or modified stem. Modified word to ami agoteও পঢ়ি আহিছো. In case of morphology we have studied, right? We have talked about the different types of modification of stems, modifications of roots, etc. Right? If you remember, storage roots asile, types of breeding roots asile, etc. etc. Just to remember what we understand by the word modified. Leaf or modified leaf, by hey modified word to mane ki? A leaf is said to be modified when it conducts some other function than its normal function. তার মানে এটা লিফৰ ফাংশন কি obviously photosynthesis, right? Apart from photosynthesis হি যদি আৰু বেলেগ কাম কৰে তেতিয়ালে আমি তাক কি বুলি কম modified word to আমি ইউজ কৰিম. Similarly in case of a root also, what is the most important function of a root? The most important function is to provide anchorage or to absorb the water and the minerals. তাৰ উপৰিও যদি rootটোৱে বেলেগ কাম কৰে, right? If it conducts some other function than this normal function, that time we use the word modified, right? So modified root or modified leaf. Now the question is, it is modified for some purpose. Modified root বুলি কোৱা লগে লগে কিহৰ কাৰণে root to modify হৈছে কিবা এটা বেলেগ কাম কৰিবলৈহে root to modify হৈছে. For example, if you talk about storage root, what we remember, the root is modified for the purpose of storage, right? So তেনেকে ইয়াতো কি হয় leaf টোৱে কি কৰে, generally when we talk about leaf, the main function is photosynthesis, but in this case stamen is also a leaf kintu it is considered as a modified leaf. Kiho karne it is not conducting the purpose of photosynthesis, instead tar function to ki reproduction. That is the reason why we are using the word modified. Secondly, we have to remember certain terms called as ami etu chapter kisuman terms pam jeneke microsporophyll, microsporogenesis, microspores, thik teneke megasporophyll, megasporogenesis, megaspore. So you need to be clear about all these terms, right? Microsporophyll, sporophyll mane ki a leaf like structure that bears spores is called as sporophyll. Micro means small spores, no? What we are talking about is, I think you have heard the word called as microspores, no? Microspores mane ki pollen grains. So microsporophyll tar mane microspore thaka leaf tuk ba leaf modified leaf tuk ami ki boli kom microsporophyll. So microspores thaka leaf tuk ami ki boli kom microsporophyll. And what is that we are exactly talking about? We are talking about the stamen. Next, talking about the stamen, the parts of a stamen, everyone knows about it. It's made up of anther and filament. So just to have a rough idea about the stamen, how does it look like? So let's see the stamen. So if you are talking about the stamen, it looks something like this. This is called as the thalamus. Now what is the thalamus? The seat of the flower is called as thalamus. Tar mane eta flower or jimanpur parts ase no, he gutei parts kini juntu jagar pra originate kore, tak ami ki buli kom, thalamus buli kom. So stamen to kopra originate korise, a thalamus to lagi thakibo, right? So ariate tomar stamen uthakibo, tomar petals bilaku thakibo, sepals bilaku thakibo. So this is called as the thalamus. Thalamus usoro juntu part of the stamen lagi ase. This is called as what? Proximal part. The part that is away from the thalamus is called as the distal part, and we know the stamen is made up of two parts, to be precise, that is anther and filament. So we have the anther and we have a tender, or we have a slender thread-like structure, that is what is called as filament. So ekhini pra amar ki idea hoy that how does a stamen look like. Now some points about the anther. So anther kisman points ami janibo lagibo. Anther can be of two types. Monothecous and dithecous, right? What is monothecous? Diagram to akile clear hobo. So this is a monothecous anther and this is a dithecous anther, right? So jedi monothecous anther ami ki mon rakhibo lagibo, it has one lobe, a lobe too, it has one lobe, has two microsporangium. So it is called as bisporangiate. This type of an anther is generally seen in the family Malvaceae, and one example of Malvaceae is hibiscus, or it is also called as China rose, right? So anthers, ami ki jano, anther filament are the two important parts of the stamen, and when we talk about anther, one other information that we know is it can be either with one lobe or it can be with two lobes. If the anther is with one lobe, then it is called as what? It is called as monothecous. Or jedi anther to enike duta lobe ase, etu eta lobe, this one and this one, then it is called as a bilobed anther. Terms to be remembered is, when we are talking about monothecous anther, we have to remember the word bisporangiate, right? Aru eta word paiso no, microsporangium. Olop agote ami ki word paisilo? Microsporophyll. Sporophyll, fill word to thakile, what we understand, we are talking about that leaf-like structure, and so that is what is called as fill. Now we are using a word called as sporangium. What is sporangium? The house of the spore is called as sporangium, right? So this is the house. Tamane if you are talking about a leaf like this, ekonok jedi ami microsporophyll buli ko, ekonok jedi microsporophyll buli ko, yate juntu spores bilak juntu jegat thakibo, tak ami ki buli ko? Microsporangium, right? And inside the microsporangium ki thakibo? Microspores. This is a diagram just to understand the differences, or we understand the meaning microsporophyll, microsporangium and microspores, right? So yate ami ki koisu je junbilak single lobe by unilobed anther hoy no, tahator ki thake je they are said to be microsporangiate. Why they are called as, sorry, they are called as bisporangiate. Why they are called as bisporangiate? Because they have two microsporangia. So jetia ami stamenor kotha koisu, stamen dal ki hoy? Male reproductive part, right? Gutei stamen dal male reproductive part, ar stamenor bhitorot duta ba stamen or duta part ase, that is anther and filament. And in the entire structure, what is the most important part from reproduction point of view? Right? Filamental important, anther to important, reproduction point of view. Karon reproduction, kuntu type of reproduction kotha koi aso, we are talking about sexual reproduction, and in that case, what is the most essential requirement is the two gametes, right? So the point that is most important is we have to talk about the male gamete. Male gamete, or it is also called as the pollen grain, no? Pollen grain or bhitorot ki thakibo male gamete thakibo, pollen grain to kot thakibo antherot thakibo. So stamen or sobotse important part to ki we are talking about that pollen grains, right? And those pollen grains will have one name, and that is what is called as microspores. Hey pollen grain bilak ba microspores bilak juntu area ba juntu structure or bhitorot thake tak ami ki buli kom microsporangium. So if the anther is single lobed, it will have 2 microsporangium, and so we are using the word bisporangiate. Similarly, we'll come to this type. The second type of anther that we see is something called as dithecous anther. In case of dithecous anther, the information that we know is it has 2 lobes. Agortot kiman asile? It had 1 lobe. Yate kiman ase? It has 2 lobes. Next, it has 4 microsporangium. It has 4 microsporangium. So we will be using a word called as tetrasporangiate. Agortot ki asile? Bisporangiate. Etia ki paiso? Tetrasporangiate. And another important point is, it is the most common type seen in angiosperms. So whenever we talk about this anther, or whenever we talk about the different lobes of anther, or which type of anther we'll be more talking about, we will be talking about this dithecous anther. Dithecous anther bikhoye ami details of kiho karone porhim, because we are talking about the flowering plants, and the flowering plants are nothing but angiosperms. So in maximum of the flowering plants, ami kenuka dhoroner anther dekha pao, we always see the presence of dithecous anther. That does not mean monothecous nathake, monothecous thake, kintu most common kuntu thake dithecous. So when is an anther said to be dithecous? When it has all these characteristics. Aru jodi monothecous hoy tetiahole you have these characteristics. Now the point to be remembered is, if an anther is monothecous, then it is considered to be bisporangiate. If an anther is dithecous, it is considered to be tetrasporangiate. Tetrasporangiate mane ki je 4টা microsporangium thakibo, aru hei 4টা microsporangium or bhitorot ki thakibo pollen grains bilak thakibo. So, 8 point or 8 point monot rakhiba. A most common type kuntu dithecous. Yate eta point janibo lagibo, eta extra point. You have a plant called as Arceuthobium, right? Arceuthobium ki janibo lage je it is the smallest dicot parasite, right? And the anther is monothecous and monosporangiate. Right? So Arceuthobium buli adal plant ase, he plant dalok ami ki koise, it's an example of a parasite. Or question hua hai smallest dicot parasite or naam tu kua, so it's Arceuthobium. What is unique? The unique point is monothecous is a word we have already understood, right? Monothecous ketia kom jetia anther to 1 lobe thakibo. When it is monothecous, generally we will be using the word ki bisporangiate. Okay. So the important point about Arceuthobium is monothecous. Monothecous hole ami ki jano je single lobe. Ar single lobe jetia hoy tetia hole ami kuntu word use korisilo bisporangiate, kintu yate ami ki word use koriso monosporangiate. So whenever an anther is single lobe, the word that is related is, or what is the number of microsporangia that it has, it always has 2 microsporangia. But this is an example where the anther is single lobe, but the number of microsporangia that is present is 1. So we are using the word monosporangiate. Monothecous bisporangiate or monothecous monosporangiate. Are in common ami ki rekha pao khodai dithecous tetrasporangiate. I think we understand till this part, right? Next is ekhini hol some points about the anther, because we know the stamen has 2 main parts, that is the anther and the filament. Next, some informations about the filament. filament or key information janibo lagibo je it is something that is long, slender, right, and almost thread like. We have to read something called as fixation of anther. So yate ketaman diagram akim sama. So fixation of anther jeta filament poru no stamen or jigetu part. So filament or logot ki log lagi thakibo. Obviously the anther would be fixed on the the anther would be fixed on the filament. Now how is the anther and the filament fixed? According to that point of view, we have certain terms to study. And what are the terms? Ki dhoroner anther, this is called as basi-fixed anther, right? This is called as dorsi-fixed. This is called as adnate, and this one is called as versatile, right? So anther or filament keneke log lagi ase, on that basis we have certain terms, that is dorsi-fixed, basi-fixed, adnate or versatile. Basi-fixed ami ketia kom when the filament is attached to the base of the anther. Etu ki hoy anther, right? Aru edal ki hoy filament, right? A filament dal kot lagi ase to the base, and so this is called as basi-fixed. Similarly, this is the filament and this is the anther, and where is the filament attached? It is attached to the dorsal side of the anther, or it is attached to the back side of the anther. So accordingly we have a term that is called as dorsi-fixed. Similarly, we have something called as adnate. Adnate, if you noticed, a portion to a black portion, shaded portion akisu no, it has been extended, right? And does anyone remember what is this portion called as? Etuk ami ki boli kom? Anther. Edalok ami ki boli kom? Filament, right? So a majo part khinik ami ki kom? Stamen edalor hodai duta part thake no, anther or filament. But have you heard the word that is called as connective? Right? So in a stamen you have another part that is called as connective, and what is the work of the connective? To connect the anther and the filament. And what is the connective? It is nothing but a mass of sterile cells, right? Stamen dalor gutei khinite kisuman fertile cells thake aru kisuman sterile cells thake. So sterile cells boli kole connective dal is a mass of sterile cells. A fertile cells kud bilak obviously anther bhitor jun bilak cells ase he bilakok ami ki boli kom? Fertile cells, karon those are the cells that are going to produce the what? Pollen grains, right? So accordingly je yate ki hobo etur condition ami ki koy? Adnate condition. Adnate condition if you see that ki hoise a connective dal no ekdom upor loke ase. So gutei majot connective dal ase, epine eta anther log lagi ase, epine eta anther log lagi ase, and this condition is called as adnate. ERA details, I think if you remember, we have done this in the morphology class also. This is just for the revision purpose. What is important from this point of view? Karon ami ki pori aso? Reproduction, and whenever we are talking about reproduction, we have to talk about pollination also, right? So from pollination point of view, one question that is asked from this portion, that is the reason why we have discussed this. We have the next type that is called as versatile. Versatile anther. So more kenuka hobo, if you notice ekhini ki boli koise anther and this is the filament. So what is the characteristic of this type of a condition is je yate ki hobo enuka dhoroner jonbilak anther thakibo no, he anther bur, suppose this is a filament, ekhinite ki hobo anther to at the tip, like so anther to thakibo, anther just eta point othe filament dal log lagi thakibo, and what will happen because of that is whenever the wind come, the entire anther can move in a round condition, right? So what I have shown is with the help of the arrowheads. So jetia bota ahibo tetia hale ki hobo e gute anther kini, not necessarily the filament need to move, filamental move nokorileo anther to ki hobo move koribo paribo, kiho karone, because that anther is attached or that filament is attached just to the tip of the anther, and that is what is called as versatile, right? One characteristic of versatile is it is something that is very common in case of anemophilous flowers. So question to ahe anemophilous flowers or kenuka dhoroner anther thake ba kenuka dhoroner filament thake, or what condition you can see the versatile condition, right? So anemophilous ki asile, those plants that are pollinated with the help of wind is called as anemophilous. So these are the informations about the filament. Some information about the anther we have discussed. Now we have discussed some information about the filament. What is left with is something called as connective, right? Connective ki eko besi monot rakhibo nalage, sterile mass of tissues, right? Next, from reproduction point of view, what is the most important structure? The most important structure is always the pollen grains, and where will you find the pollen grains? It's always inside the anther. So if you want to study or if you want to know how does the development of the pollen grain takes place, or where can you see the pollen grains precisely, what we need to study, ami kuntu part poribo lagibo pollen grains to sabol lagile, obviously we have to talk about the anther, right? Anther to jodi ami study koru tar bhitorte ami ki dekha pam, pollen grains dekha pam. So in order to study the interior contents of an anther, what we need to do, obviously we have to cut a TS, right? Etia ami sam anther or bhitorto no kenekua. So if we want to study the development of the pollen grains, now how does the pollen grain get developed? We know anther bhitor pollen grains ase, kintu he pollen grains blak korpra develop hole, which cell is responsible to form the pollen grains, right? So all these things if you need to study, we have to cut a TS of an anther. What we have written is we have cut the TS of a young anther and not a matured anther, because if you take a matured anther, everything or you cannot see the process of development, right? Already to be anther to dekha ba tar bhitor ki dekha baba pollen grains blak already form hoye ase, but if we are talking about a young anther, we can see the process of development of the pollen grains, and that is what we should know in this chapter, right? TS of a young anther. So how does it look like? So we are talking about this dithecous anther. Why are we considering the dithecous? Karon kyo dithecous to sobotse common hoy no. So dithecous anther jedi tumi TS kora no, TS of a dithecous anther, yet as it will half period, then you will see a structure like this. Eneke kisuan sarita lobe thaka structure ga dekha paba. Enekwa sarita lobes kiho karone ba sarita enekwa curve kiho karone ase, because we are talking about the dithecous no. A portion to jodi kati dia iar pra eta aru iar pra duta aru epine jodi kata iar pra eta aru iar pra eta. So ultimately iar jetia TS korim, so we are going to see a structure like this, right? A structure dot ami ki dekha pam, we will see outer layer of cells, or outer layer to kami ki boli ko, epidermis, right? So এনেকে এটা লেয়াৰ পাম আৰু লেয়াৰটোৰ ভিতৰত আমি কি দেখা পাম যে ইয়াতে কিছুমান সেলস দেখা পাম right So when we cut a TS of the anther, what we are going to see is we are going to see certain types of tissues or we are going to see certain types of cells, and these cells or this group of tissues are limited or has a boundary, and that boundary is called as epidermis. So this is a rough diagram that we are going to see because of the TS. So enukha eta lobe structure dekha pam tar bitor bahiror layer tuk ami ki boli kom epidermis, and epidermis or inner side ole ami ki pam eta hypodermal layer, and this layer is called as archisporial layer. এখিনলৈকে ক্লিয়াৰ হৈছে নে নাই? সো আমি যদি ইয়াং এন্থাৰ এটা টিএস কৰো তেতিয়াহলে টিএস কৰিলে আমি কি দেখা পাও কিছুমান টিস্যুজ দেখা পাও ন আৰু যেতিয়া টিস্যুজ বিলাক লেয়াৰ হৈ থাকে এটাৰ ওপৰত এটা আউটাৰমোস্ট লেয়াৰটোক আমি কি কওঁ the term that we use is always called as epidermis, right? So similarly in this case also we are going to see the outer layer of tissue that is called as epidermis. Epidermis or bhitorer pine but towards the inner side of epidermis again we are going to see certain types of tissues, and this is what is called as the archisporial layer, right? Etia ei cells bilakor porai pollen grains bilak develop koribo keneke develop koribo we'll talk about, right? So when we cut a TS of a young anther, what we are going to see is homogeneous mass of cells or tissues limited by an epidermis. As I said before, outer layer to ami ki boli kom epidermis. Below the epidermis, below it ami ki pam the hypodermal layer called archisporial layer. Right? অলপ আগতে যেনেকৈ আমি বস্তুটো আঁকিছিলোঁ ন এনেকৈ তাৰে এইটো পৰ্চনক আমি কি কৈছিলোঁ এপিডাৰ্মিছ কৈছিলোঁ আৰু এপিডাৰ্মিছৰ এই ভিতৰৰ এই চেলছবিলাকক আমি কি বুলি কৈছিলোঁ যে দিজ আৰ কল্ড এজ এইবিলাকক কি বুলি কৈছিলোঁ আৰ্কিস্পৰিয়েল ন archisporial layer, and this is the epidermis. After that what happens is in the archisporial layer 4 cells becomes prominent. 4 cells in the sense যে আমি এনেকুৱা এটা স্ট্ৰাকচাৰ আৰু এনেকুৱা এটা স্ট্ৰাকচাৰ দেখিছিলোঁ সো এপিডাৰ্মিছখনটো ইয়াত থাকিবই প্ৰথমতে কি দেখিছিলোঁ যে গোটেইবোৰ এনেকুৱা 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 গোটেইবোৰ চেলছ আছিলে ন তাৰপিছত গ্ৰাজুৱেলি উইথ দ্য প্ৰচেছ অফ ডেভেলপমেণ্ট তাত কি হ'ব এই লেয়াৰটোত এটা সেল প্ৰমিনেন্ট হ'ব সিমিলাৰলি এটা সেল প্ৰমিনেন্ট হ'ব ৰাইট এই আৰ্কিস্পৰিয়েল টিস্যুজ যিমানবোৰ আছিলে তাৰ ভিতৰত এটা সেল প্ৰমিনেন্ট হৈ বাকিবোৰ সৰু সৰু হৈ থাকিব প্ৰথমতে সব একে সমান আছিল আৰু তাৰ পিছত কি হ'ল যে এটা সেল অফ দিস লেয়াৰ will get prominent, right? So 1 2 3 4, right? এই চাৰিটাক আমি কি বুলি কম archisporial cell, right? So we'll come back to this. Cutting the TS of an anther, we will see a mass of tissues. Bahiror layer to ki boli kom epidermis. Epidermis or tolot ki pam archisporial layer. In the archisporial layer 4 cells becomes prominent and are called archisporial cell, right? So yate hol anther katilu, anther katar pisot archisporial layer dekilu, archisporial layer eta eta ki hobo je cell enlarge ho jabo ba prominent hobo egita ka ami ki boli kom archisporial cell. Next, what will happen to the archisporial cell? আমি কি পঢ়ি আছো process of development, right? So each archisporial cell, okay, ইয়াতে লিখি দিও next stepত কি হ'ব each archisporial cell will divide periclinally and form two cells. And what are the two cells? Outer primary parietal cell aru inner primary sporogenous cell. Right? So up till now what we have studied this, so a diagram to logot jodi study koro, then it will become easier. আমি কি পঢ়িছিলো যে যেতে ts কাটিম কামেই নকৰে so যেতিয়া আমি ts কাটিম ts after the ts what we will see, or because of the ts what we will see, we'll see a layer that is called as epidermis. Below the epidermis ki asile je we had that layer that is called as archisporial layer. Archisporial layer ot ki hoisile one cell became prominent and this cell was called as archisporial cell. No, etu kami ki boli kom epidermis, right? So this is the first step. What is the next step? We'll have the epidermis. This archisporial cell will now divide, right? And how will it divide? It will divide in a periclinal, or it will show a periclinal division. Now what is periclinal? Right? We have two types of division. One is called as anticlinal or eta ki boli koy periclinal. Periclinal division mane ki parallel ki bahar eta agor cellor logot parallelly je divide kore tak ami ki boli kom periclinal. We are talking about the epidermis. Epidermis of parallelly ki koribo, he divide koribo, tak ami ki boli koiso periclinal division. So eta cell asile a cell to divide kori pla duta cell form korile. তাৰ ভিতৰত এই বাহিৰৰ সেলটোক আমি কি বুলি কম প্ৰাইমাৰী পেৰাইটেল সেল আৰু এটোক আমি কি কম প্ৰাইমাৰী স্পোৰোজেনাছ সেল right I think it's understood up till here একেলকে বুজি পাইছো নে কিবা ডাউটস আছিল So enter eta what we have done is we have cut the TS, and while cutting the TS what we have seen is a group of tissues. That tissues is limited by one layer that is called as epidermis. Below the epidermis was archisporial layer, and in the archisporial layer what developed is one cell that became prominent, and that is called as archisporial cell. What happened to the archisporial cell? It divided it to form two cells. What are the two cells? Primary parietal cell and primary sporogenous cell. If you notice here, ami primary parietal cell or agot eta word use koriso no outer, ar yate ami ki use koriso, we have used the word inner. Now inner or outer, it refers to what? To understand this, we have to draw the diagram again. So when we have cut a TS, we have got a structure like this, right? Something like this. So yate ki hoisile archisporial cell to develop korisile, right? This archisporial cell will divide periclinally and ultimately will form two cells. Itu eta cell, itu eta, this one, this, and this, right? Tar bhitorot the cell that is towards the inner portion, this part is called as inner primary sporogenous cell. Our A cell to junto outer side ot ase, this cell is called as outer primary parietal cell. is the concept of inner and outer clear now, right? Okol ami ki koa nai je primary sporogenous cell ba primary parietal cell boli koa nai. We have used the word outer primary parietal cell and inner primary sporogenous cell. Now what we understand by the word inner and outer? Inner is referred to that cell which is towards the center, and outer is that cell which is towards the epidermis. Ekhinte ki asile? Epidermis kon asile no? Right? So this is the condition that we are talking about. So archisporial cell got divided into two cells, that is primary parietal cell and primary sporogenous cell. Now this primary parietal cell will divide both periclinally and anticlinally, right? So let me write it here. Primary parietal cell, no? Etu ki asile? Outer primary parietal cell. Outer primary parietal cell ki hobo? Both periclinal aru anticlinal. Tamane all direction of tahale divide koribo, and then they will form 2 to 6 layered wall of the pollen sac, right? So a layer dwe ba parietal cell dwe divide or redivide koribo, enege bahut bilak cells form kore. Cells bilak form kori belai ki form korile je pollen sac or wall khon kune form korile? It is a result of the division of the outer primary parietal cell. Similarly, we have to talk about this inner sporogenous cell also. Inner sporogenous cell will divide to form sporogenous tissues. As sporogenous tissues or ultimately ami ki bilak pam? Pollen grains. Right? Ekin loke kiba doubts ase niki? So inside the anther we have the epidermis. Epidermis at tolot ki asile archisporial layer. Archisporial layer eta cell develop korisile ba prominent hoisile, that is called as archisporial cell. That archisporial cell divided into two parts. One part is called as parietal cell and the other part is called as sporogenous cell. What did the parietal cell do? Parietal cell divided and redivided to form many layered wall, right? Aru kihor wall? Pollen sac or wall khon kune banaisile? Parietal cell. And what is the work of the sporogenous cell? That this particular cell divided and redivided to form a group of tissues, and that is what is called as sporogenous tissues. Sporogenous tissues bilake ultimately ki form koribo? Pollen grain, right? Now we have to talk something about this wall, right? Pollen sac origin to wall ase. Hey, wall khon is made up of some layers of cells, right? Kiman layers boli koiso? 2 to 6 layer. No. This pollen wall sac consists of kenuka cells ami pao. First, we'll see epidermis. Information about epidermis: single layered. Epidermis kon generally ki hoy? Single layered hoy. Function ki hoy? Protective, right? And it shrivel mane falls off at maturity. So these are the points about endodermis. Single layered. Below the endodermis ki thakibo? Endothecium. Endothecium or points single layered, right? Next has alpha cellulosic fibrous bands. A point to important question ahe alpha cellulosic fibrous bands to tumi kuntu tissues or pump ba kuntu layer or pump we will see them in the endothecium. What is the endothecium? It is the second layer of the pollen wall, right? Pollen wall is made up of many layered, or it's a many layered structure. In that types of in that wall eta layer or naam ki endothecium, or endothecium or ami ki pam alpha cellulosic fibrous bands bilak pam. Now what is the function? It helps in breaking of the anther due to hygroscopic nature. So pollen thakibo, ei pollenor ki thakibo walls thakibo no. So etuk ami ki boli koisilo sporogenous cell boli koisilo. First ami ki dekhisilu je eta epidermis asile no. Epidermisot eta cell divide korile etuk ami ki koisu sporogenous cell, etuk ami ki boli koisu parietal cell. This parietal cell divided and redivided and they form the wall of the pollen grain, right? So ei gutei khini kune form korise parietal cell e form korise, right? Ar bhitorot ki ase sporogenous cell to thakile tar bhitorot ei je wall khon form korile no yate swa already ekhon epidermis asile tothapi yate aru eta layer form koribo, yate aru layer form koribo, yate aru layer ei layer tuku ami ki boli kom epidermis kom, tar next layer tuk ami ki boli kom endothecium, right? So we are talking about the second layer no. Etu jodi epidermis hoy etuk ami ki boli kom endothecium. Or diagram to aru properly didim, then it will become more clear. As of now what we need to remember is endothecium layer ot ki thake alpha cellulosic fibrous bands bilak thake. Tar mane ei cell bilakot suppose endothecium or cell etu no tate kisman enuka dhoroner bands dekha paba ba enuka dhoroner kisman structure you see, and this bands has been formed because of the deposition of cellulose, right? So deposit hoi goise hekane kisuman ami bands dekha pam aru he bands bilako ami ki boli koisa alpha cellulosic. What is the function of these bands? It helps in the breaking of the anther. Now why is it important that the anther should break? We know the pollen grains are present inside the anther, and until and unless the anther bursts or breaks down tar pora pollen grains bila ketia ulai nahe. So anther to break koribole lagile kunubai tak help koribo laibo, and who is helping here is these fibrous bands that are present, right? Aru keneke help korise? These cells have hygroscopic nature. Hygroscopic nature mane ki that these cells has the capacity to absorb the water. Jete water absorb koribo tete anther to ki hobo swelled up hoi jabo, and as a result it will lead to the bursting of the anther and the release of the pollen grains. So this is the function of alpha cellulosic fibrous band. Now the question has not been asked that what is the function of the alpha cellulosic fibrous band. Question ki ahise the ablak bands ami kot pam. We are going to see in the layer called as endothecium. And what is endothecium? It is one layer of that sac, or it is one layer of that pollen wall, right? Pollen grain wall khon eta layer. Aru eta question related to this is alpha cellulosic fibrous band is absent in A point 2 important. Absent in. This is absent in hydrophytes. Right? So in a apart from hydrophytes sob bilak plants or ki thakibo a bands bilak thakibo kintu hydrophytes are na thake. What is the reason behind? Because we know if a plant is a hydrophyte they do not need a specific cell to absorb the water. They can absorb the water from the entire body surface. As a result specific bostu tahatok nalage, right? So hydrophytes are absent, baki bilak plants of kintu thakibo. So please pay interest to this point. Next, epidermis or tolot ami ki palu? Below the epidermis is endothecium. Now we'll talk about the third layer below the endothecium. Ami ki dekha pam? Middle layer. Next is informations about the middle layer. Two to three layered. Yar agolo kya ami jeta dekhisile epidermis single layered, endothecium was also single layered, but middle layer is made up of 2 to 3 layered, and these cells they are made up of loosely arranged parenchymatous right, and they are very short-lived. And whenever we are using the word short-lived, eta word janibo lage ephemeral. What is ephemeral? Short-lived is called as ephemeral. Okoman dino kare ziyai thake eta kami ki boli kon ephemeral. So pollen saccor jonkon wall hoy, hey wall kon single layered or multi-layered? It is obviously multi-layered, right? And what are the layers that you can see? Ekdam uppermost we see the epidermis. Below the epidermis is the endothecium. Inner to the endothecium is the middle layer, and lastly we have a layer that is inner to the middle layer also. Next we have the innermost layer that is called as tapetum, and this is very important, right? So layer bilakor ki asile epidermis, endothecium, middle layer, a middle layer tolot ki ase tapetum. Informations about the tapetum. Ami ki jano? Innermost layer, right? It surrounds the sporogenous tissues. It has a large nuclei or a thick cytoplasm, right? Next is some words that you need to remember: multinucleate, right? Next is the cells of the tapetum are generally multinucleate. Multinucleate mane ki? Generally what we know is each cell has a single nucleus, but if a cell has more than one nuclei, it is called as multinucleated condition. So question might be asked as multinucleated condition is a characteristic of dash cells. So multinucleated condition is a characteristic of the tapetal cells. Next is they are an example, or they show polyploidy, right? Next they have high DNA content. So tapetal cells bilak birat important. Ekan tare information ami okoman janibo lagibo. And lastly, function of tapetum ki hoy? Provides nourishment. And nourishment to whom? To the developing sporogenous tissues, or in short we can say they provide nourishment to the developing pollen grains, because we know that from that cell, that archisporial cell that we have talked about, duta cellole divide korisile, eta asile sporogenous cell, aru sporogenous cell ki form koribo? Pollen grains form koribo. So pollen grains bilakor obviously they will need food, and who is going to provide the food? It is always the tapetum that is going to provide the food. So these are the informations about tapetum. Tar bitoron important ki multinucleate important, polyploid important, high DNA content is also important, obviously providing nourishment is also important. So ekini whole points about the tapetum. Now okay, up till now ami kenukata structure dekhisu. So I will draw a structure. Sorry, এতিয়ালৈকে আমি যিখিনি পঢ়িছোঁ, let's put it in the form of a diagram. Right? So, etu ki asile? The epidermis, right? Epidermis or tolot, what we have seen is epidermis or tolole ami ki ba innermost to the inner to the epidermis we have seen a layer that is called as endothecium. So, this green portion represents what? Endothecium. Right? Below to the endothecium ami ki dekhisilu, we have seen loosely arranged parenchymatous cells. Or kiman layer? 2 to 3 layer. Right? And below this we had the these are nothing but the tapetal cells. They are pyramidal in structure, right? Sorry. Right, our inner to the pyramid we have the sporogenous tissues. So, this is what we are talking about, right? So, what does this diagram tells us? We'll mark the different layers. This layer is called as epidermis. Inner to the epidermis we have endothecium. This is called as middle layer, right? And this one is called as tapetum. And inner to the tapetum what we have? Sporogenous tissues. Right? So, ultimately, etialake ami jikhini bostu porilu no, whenever we cut the TS of a young anther, we see certain groups of, or we see certain layers of tissues, and how are the layers arranged? The layers are arranged in the form of first layer to epidermis cell, tar pisot ki asil hypodermal layer. Hypodermal layer was also called as archisporial layer. In the archisporial layer, you had a cell that is called as archisporial cell or archisporium. Archisporium got divided into two cells, parietal cell and sporogenous cell. The parietal cell formed all these layers of this pollen grain. A sporogenous cell or surrounding layer to nam ki tapetum. So, we have written the point, tapetum is considered to be the innermost layer, right? And it surrounds the sporogenous tissues. So, ebilak jodi sporogenous tissues hoy, tak kune surround korise? Tapetum surrounds korise. It is a state, or these are examples of multinucleated condition. They show polyploidy and they have high DNA content. And what is the most important function of tapetum is to provide nourishment. Now, we have to talk about something called as polyploidy. What is polyploidy? Haploid, diploid, polyploid, no? If suppose a cell has one set of chromosome, it is called as haploid. Two sets of chromosome is called as diploid, and it has more sets of chromosome, it is called as polyploid. Since it has more sets of chromosomes in one single cell, obviously tahator DNA content to besi hobo, right? So, give an example of a cell that shows polyploidy, you could always give the example of a tapetum. Now, what is the reason that they have high DNA content? Normal cell attack tapetal cells bilakor amount of DNA that is present is always more. There should be certain reasons for that, right? So, what are the reasons? I mean, the reasons can be some. So DNA content or basi DNA thakar reasons ki it may be due to endomitosis, right? It can be due to formation of restitution nucleus, or it can be due to polytene condition or polytene condition. So DNA content code besi thakibo, tapetal cells bilakot besi thakibo, kihor karone besi thakibo. So these reasons are considered to some of the explanations for the increase in the DNA content. The first one is what they have said is it might be because of endomitosis that the DNA content has increased. Now what is endomitosis? Endomitosis, endo means what? Inside, right? Aru exo mane ki outside. So endomitosis is a process of mitosis in which all the stages of cell division occurs within that nuclear membrane. Normal mitosis and endomitosis are different ki. Normal mitosis ami janu je jetiai arambho hoy no, the starting of normal mitosis is always prophase, and what is the first characteristic of a prophase is that the nuclear membrane will start getting degraded, right, by disappearance of the nuclear membrane. Nuclear membrane kon disappear koribo aru tar pisot cell division or baki stage bilak tat hoi thakibo. Kintu in case of endomitosis ki hoy, the nuclear membrane does not disappear and all the other functions occur within that nuclear membrane, and that condition is called as endomitosis. So scientists are of opinion that because of endomitosis the DNA content of the tapetum has increased. So what is endomitosis? The nuclear membrane kon remove nokorake mitosis code hoy ase within the nuclear membrane. The next point is formation of restitution nucleus. Restitution nucleus, eta nucleus or condition attack ami restitution nucleus boli koiso. What is the condition or what happens here? What happens is in such type of a condition ki hoy that normal mitosis soli thake, thik ase, normal cell division soli thake up till the point of anaphase. Okay, we'll go back to cell division now. Whenever we talk about cell division, ketaman stage ase no, jeneke if you are talking about the first stage, that is prophase, prophase followed by metaphase, tar pisot anaphase aru tar pisot telophase. What happens in prophase? Nuclear membrane gets separated, gets degraded, nucleolus disappear kore, right? Chromosomes bilak ki hoy scattered in the cell hoi jay, right? Those are the characteristics of prophase. Tar pisor stage to ki boli koy metaphase. Metaphase ot ki hoy, that all the chromosomes they start aligning themselves in the equatorial plan. So jiman bur cellot tumar chromosomes bilak scattered hoi asile, ei guti chromosomes bilak ki kore, eta equatorial planor properly arrange hoi jay. That is the characteristic of metaphase. Followed by metaphase is anaphase, no? So suppose we are talking about this one as prophase. Protomote nuclear membrane thakibo aru yate chromosomes bilak thakibo, jetia nuclear membrane degenerate hoi jabo tetia hale tat ki hobo cell to chromosomes bilak separate hoi jabo. Etuk ami ki boli koiso? Prophase. Prophase followed by ki metaphase. So what happens in metaphase? That you have this cell and you have the chromosomes that start aligning in the equatorial plan, right? So mazot ahibo tak ami ki boli ko? Metaphase boli ko, right? And after metaphase ki thakibo? Anaphase. What happens in anaphase? Je a chromatids bilak separate hoi jabo, kisuman yale jabo aru kisuman yale jabo. So you have this, and so anaphase of ki hoy, the chromosomes they will move to the two respective poles, and after anaphase what we have is telophase. What happens in telophase? We have this jun bila chromosomes asile tat ki hoy jabo je nuclear membrane kon ako tahator form koribo, right? So these were the basic steps of cell division. In case of a restitution nucleus, enukwa dhoroner process ot ki hoy je anaphase or loke process to ekei thake. Prophase ot ji hobo lage hebilak hoi jabo, metaphase ot ji hobo lage hebilako hoi jabo. Anaphase or pisot কি হব লাগিছিলে যে chromosomes বিলাক দুটা polole গুছি গল. That is the characteristic of anaphase. After anaphase কি হব লাগে যে গুটে বিলাক chromosomes তাহাতৰ কি কৰিব লাগে, nuclear membrane খন আকৌ develop কৰিব লাগে. So ultimately what is the number of nuclei we'll see after anaphase? It is always 2. But in case of a restitution nucleus, what happens is no doubt the chromosomes move to the two opposite poles, but they remain surrounded by the same nuclear membrane. So restitution nucleus condition to kenukwa hoy যে এনাফেজ হ'ল এনাফেজত মুভ কৰিলে ৰেসপেক্টিভ পোলছলে মুভ কৰি পেলাই তাহাতে কি কৰে the same nuclear membrane খনেৰে সাৰাউণ্ড হৈ থাকে surrounded by the same nuclear membrane and such type of a nucleus is called as restitution nucleus. A restitution nucleus etia swa ekhon membrane ase jehetu it will be considered as one nucleus. If it is one nucleus, then the number of chromosomes would be more and that would result in the increase in the DNA content. So they were of opinion, scientists were of opinion that because of restitution nucleus might be the DNA content in the tapetum increase. Kihor karone increase hoise ebilak hol ki different reasons. Endomitosis of karneও increase hobo pare, restitution nucleus form korise he karneও hobo pare, and lastly we have something that is called as polygyny. So they were of the opinion that polygyny